Strombaum. I'm the Vice President overseeing the program, and I'll give you a brief intro before I pass it on to my colleagues, Javi Cruzan and Jun Shu, who will then bring on our 4 panelists. So at any point in time, please feel free to submit questions to our panelists. You'll see the Q&A feature. You can put your question in, and we're going to monitor this box. We'll answer a few questions probably in real time, and then we'll bring questions to our panelists. I also want to let you know we're recording this event, and of course, we'll make the recording and additional information on our program available to you after this event. So our TAP program is really one of the first venture philanthropy programs in the biomedical space. It was created originally in 2007 by our CEO, Lou DeGennaro. And it's a program that conducts investments, equity co-investments in both private and public companies, and focusing really on companies that are developing truly novel, potentially groundbreaking therapies, companies worldwide. These mission-driven investments do not have a financial component, and more importantly, there's a collaboration involved that really helps us work with the company on making sure that the development of these agents can provide the maximum benefit to patients at the end of the day. The program has invested over $130 million to date. We're actively investing as we speak, and we're very proud to say that our funding has contributed to 4 approved therapies, among them Vyxeos, a novel liposomal formulation of cytotoxic therapy for AML patients with a particularly poor prognosis, such as secondary and therapy-related AML. We're really excited that this therapy now, since 2021, is also available to pediatric AML patients. And then, of course, Yescarta, one of the first 2 approved CAR T therapies, the first one approved for large B-cell lymphoma, a real breakthrough with CAR therapy for many of our lymphoma patients. And beyond that, of course, now we're seeing the impact with approvals in multiple myeloma as well. And then Elzonris, a really good example for the first approval of a very rare indication in blood cancer, a dendritic cell neoplasm, in 2018, really the first approved agent there. And then we're also going beyond the novel approvals of novel agent in terms of helping to expand indications for agents that are already approved. And a good example for that is Copiktra. It's a dual PI3 kinase inhibitor that had already been approved for SLL and CLL, as well as for follicular lymphoma. And our work contributed to a clinical trial that supported the inclusion of this agent in the NCCN guidelines for forms of peripheral T-cell lymphoma. On the right-hand side, here we give you a snapshot of companies who have partnered, who are partnering with our TAP program. And I just want to highlight here again that we're pretty comprehensive. This includes agents at all stages of development, from preclinical here on the outside through phase 1 all the way to registration-directed trial. It includes all indications within blood cancer, and in this we include, of course, leukemia, lymphoma, myeloma, myelodysplastic syndrome, and also myeloproliferative neoplasm. So we're trying to be very comprehensive here. And of course, we are also including all modalities. We understand that there's a need for different modalities, opportunities, also based on what patients need. So we'll find small molecules, biologics, cell therapies, novel approaches involving gene therapy, editing. It's a very, very broad approach. Now, in our investment strategy, we really focus on those companies and those programs and candidates that we think can make a dent in the treatment landscape. So we're not just looking at what's needed today. We're looking at what are the unmet needs that are emerging. With new approvals, sadly, also, we see still relapses, and we see a need for novel agents, even in the post-CAR T world. So we carefully look at these companies, at these opportunities, and we're looking for true innovation, first-in-class assets, platforms that have the potential to deliver multiple therapies for multiple indications. Our due diligence is a stage-gated process. You can see a brief outline on the right-hand side. We take advantage of our ample connections in the academic, scientific, and clinical world, as well as in the business world, to support us here. And it's a quite comprehensive process that's not just looking at the science and the clinical development plan, but also at the seasoning of the company, the management team, intellectual property, and finances. Now, today specifically, it's a real pleasure that we'll be speaking with our European partners. As I mentioned, we have a worldwide reach, and our first European partner company was Affimed, and we're especially pleased to welcome Adi Höss today during our discussion. We could speak about his experience working with TAP. You see some of the early companies also in Europe, including Kiadis and Argenix. You will be hearing from Fair and Raivu and Imcheck, and I want to highlight, as of yesterday, we are really excited to welcome a new European tech partner with BioInvent, a company that has an approach to develop some really novel immunomodulatory antibodies and has 2 clinical stage assets that are being developed. So at this point, I would like to call on my colleagues, Javid and June, to please turn on their cameras and get us started with the panel discussion.
Thank you, Laura. I'm June Xu, Executive Director and TAP Lead for the Therapy Acceleration Program. I have been with the TAP program for over 11 years, and I'm very proud of the achievement of TAP. Together with Javid, we will be moderating today's panel discussion. Javid, please say a few words to introduce yourself.
Thank you, June, and thank you, Laura. I'm Javid Furzan. I'm the VP of Business Development and Strategic Alliances for LLS. I'm the business lead for the TAP program, and I've been with LLS for just over 6 years now, and pleased to have worked with executive management and others to position TAP as a very collaborative, focused program to help accelerate innovative new therapies. Without further ado, I'd like each of our panelists to turn on their cameras and microphones, if not already done, and turn it back over to you, June.
Okay, thank you, Javid. Welcome to all the panelists, and thank you for joining us today. Let's start by having each of you to introduce yourself, your company, and your focus in blood cancer space. I go with the alphabetical order by company name. So I start with Affimed.
Thank you, June. Indeed, thanks a lot for inviting me to this panel. Pretty excited to be able to speak here. My name is Adi Höss. I'm the CEO of Affimed. We are a clinical stage immuno-oncology company, indeed headquartered in Germany, in Heidelberg, but we have offices in New York City and in Philadelphia. And since 2014, we are a NASDAQ listed company, indeed performing a listing straight on NASDAQ without any European listing. I myself, have a PhD and an MD and have worked in the biotech field for many years, almost for my entire professional life, founding companies, starting early with companies such as MorphoSys, a well-known today therapeutics, antibody therapeutics company, or Jerini, a company that has developed a product for rare diseases called hereditary angioedema. Today, this product is detailed by Takeda. From the beginning, my focus was on, when I joined Affimed, which is about 10 years ago, from the beginning, my focus was on transforming and indeed elevating the company from a fairly early stage, almost academic setting, having excellent preclinical engineering, just about 20 people, to a now mid-stage clinical development pioneer of the innate immune activation for cancer treatment, an area today that's pursued by many companies, but when we started, there weren't that many around. Today, we're about 170 employees, and indeed have a significant number of industry's most talented scientists, entrepreneurs, and seasoned leaders. It's indeed a particular honor for me to be here today, in particular as Affimed and LLS have a very long history together, including a study partnership on the lead program that's been of very great importance of our company.
Thank you.
Can I speak? Should I say something about Affimed?
Yeah, just briefly,
Yeah.
yes.
So Affimed itself is following the idea to activate innate immunity through targeting natural killer cells and macrophages. We have a platform that gives us access to bispecific antibodies that have shown to possess very high affinities to the innate cells, so macrophages and natural killer cells, that can really take these cells and move them to the cancer cells, which eventually leads to a destruction. We've obviously learned that this can be achieved with patients' own NK cells and macrophages, but very often the immune system of such patients is indeed very weak, and has also low numbers of cells. That's why we have looked into how we can enhance the efficacy of these single agent molecules, and have identified combination opportunities with PD-1 antibodies, but also most recently the combination with natural killer cells, and these are allogeneic natural killer cells. This all was exemplified and achieved by AFM13, as I said, a drug that has been supported by LLS now over many years. We have concluded studies in Hodgkin lymphoma and peripheral T-cell lymphoma, and indeed in both indications, patients are of a significant need of not only receiving effective therapies, but it's also time to bring safer therapies, and that's what the innate cell activation is promising. Monotherapy is indeed delivering very nice single agent efficacy. We've just published data at ASH or around ASH on AFM13 in peripheral T-cell lymphoma, and the results were very similar, looked even a little better than for those drugs that are already on the market for peripheral T-cell lymphoma. Still, we have discovered something now far more exciting. It's combining AFM13 with NK cell. That's a collaboration that's ongoing with MD Anderson. They provide Coplast-derived NK cells, and we've again reported at ASH most recent data. We've now treated 41 patients, again heavily pretreated patients. The last therapy, patients didn't respond, so very, very sick patients, also sick from disease, probably also sick from getting treated with toxic therapies. We learned that the therapies very safely to be applied. Patients are indeed mostly treated outpatient, and what was very stunning were the response rates. So we saw above 90% of patients responding, and above 70 are having a complete response rate. Okay, now it became time to think about how this combination can be brought to market. We're now identified a large manufacturer of natural killer cells, a company called Artiva. We signed this relationship and now are looking forward to initiate a registration-directed study. So we're in the process of filing with FDA soon. We have a second program in liquid tumors, addressing one of the key areas that you just described of high medical need, as such AML and MDS, very significant need. It appears that those patients may respond to NK cells much better than to T cells. As such, we're pretty happy now to also initiate a clinical study with AFM28, a CD123 targeting innate cell engager. Thank you very much.
Okay, thanks, Adi. Let's move to Farron.
Thank you very much, June. Thank you for the invitation and the opportunity to Farron to participate in this panel discussion. So my name is Marie-Louise Fjellskog, and I'm the Chief Medical Officer at Farron. So Farron is a clinical stage pharmaceutical company focusing on tackling difficult to treat cancers and inflammation with precision immunotherapy. Farron was founded in 2006 as a spin-out of Turku University. So we are headquartered in Turku, Finland, but we also have an office in Switzerland, and we do have an increasing presence in the US with an office in Seaport in Boston. We are listed on AIM London since 2015, and we also have a listing on Nasdaq First North in Helsinki since 2019. The main focus today, clinical development at Farron, is on the development of Bexmarilimab. Bexmarilimab is an IgG4 antibody targeting Clever-1. Clever-1 is a novel macrophage checkpoint. By targeting Clever-1, we can polarize the immune suppressive M2 macrophages into immune activating M1 macrophages. So we get an immune activation and we can fight the cancer. We started our development in solid tumors, and we have data from more than 200 patients showing that single agent Bex is well tolerated, and we have promising efficacy data. However, now we are focusing on blood cancers. One of the rationales for going into blood cancer is that, in solid tumors, we're indirectly targeting the cancer by targeting the macrophages in the tumor microenvironment. With myeloid malignancies like AML, MDS, the cell that we target, the myeloid cell, is the immune cell, but also the cancer cell. So it's a more direct way of targeting the cancer, with the cancer cell expressing the Clever-1. We initiated a study last year, a phase 1/2 study, where we add Bex to standard of care in AML and MDS, and I hope to be able to share some data about that later on. Thank you.
Thank you. Let's move to ImCheck.
Hey, June. Hi, all. This is Pierre. I'm the CEO of ImCheck Therapeutics. We are a privately held biotech company based in the south of France. And we've been incepted in 2015 around the idea that we could develop antibodies against almost every member of the butyrophilin superfamily. Butyrophilin is a understudied family of receptors comprised of 14 genes. And our scientific founder, Professor Daniel Olive, who resides in Marseille, and that's how we came to found the company here, is around the discoveries that he's made, and notably around butyrophilin 3A, which seem to be playing a very critical role in the activation of gamma delta 2 T cells. So ImCheck is centered in its first clinical program around activation of gamma delta 2 T cells in the context of solid tumors as well as hematologic malignancies. And we've became a clinical organization in Q1 2020. Have now established very strong safety evidence, not only in monotherapy as well as in combination with different settings, and are just started the expansion phases of our study with very interesting initial responses, partial and including a complete metabolic response in solid tumors at this stage, but very interesting and encouraging results in AML as well as lymphomas. We've raised our Series C about 6 months ago, about $100 million, which is actually a record, a new record for a private biotech company in France in the therapeutic area. And there was the opportunity for us to welcome LLS among our new investors, which, and I'm sure we'll have a moment to talk about it later, brings us a lot more than just the money, but a fabulous support and networking capabilities. So that's ImCheck as a whole.
Thank you. Let's move to Raivu, and also another long-term partner for TAF.
Good morning, ladies and gentlemen. My name is Paweł Przewięźlikowski. I'm the Chief Executive Officer of Raivu Therapeutics. I'm also the co-founder of the company. I've been with Raivu for the last 15 years. Raivu is a publicly listed small molecule oncology drug discovery company with 2 drugs currently in the clinic, Raivu 120, which is a lead program. It's a first-in-class dual CDK8/19 inhibitor, currently enrolling patients in a phase 1 study in acute myeloid leukemia and high-risk MDS, and separately in a solid tumor study. We also have developed another drug, which is currently in phase 2. It's a first-in-class dual PIM3 kinase inhibitor, globally partnered with Menarini. Currently, the phase 2 study is focused on IDH-mutated AML patients. Behind Raivu 120 and Cell24, Raivu has a broad pipeline of programs at the discovery stage and preclinical stage, in 2 platforms, synthetic lethality and immuno-oncology. So regarding RIVO120, obviously the main mechanism of action is targeting RNA transcription and specifically mediator complex through CDK8/19 inhibition. And we are very pleased to see single agent efficacy of the drug, a complete response, several platelet responses, erythroid responses. So we believe that this drug has already proven strong potential, and we are planning to bring it into phase 2 studies later this year. Our collaboration with the LLS has dated since 2017, and we are obviously a top funding recipient. And in our fundraising last year, in which we raised more than 50 million euro from the financial investors, LLS has also participated. So we can now welcome LLS not only as a kind of top partner, but also an investor in RIVO, and we are very happy about it. And I hope to tell you more about RIVO120 and our collaboration with the LLS later in the panel. Thank you.
Thank you, Paweł. Thank you all. Thanks. Before we go on, I'd like to remind the audience that if you have questions, please use the Q&A box. As Laura pointed out, Affimed was TAP's first European biotech partner, and TAP became very interested in the novel technology platform, in particular AFM13, which is a first-in-class innate cell engager. So LLS and Affimed entered into a research agreement in 2013 and to support the clinical development of AFM13, and that this agreement, was before Genentech signed agreement with Affimed. And so Adi already told us all the latest exciting news about AFM13. But I remember that, you, Adi, you personally were very involved in the whole due diligence process from the very first meeting at ASH 2012. So I'd like to ask you if you can go down the memory lane, share some of your memory of those early days. So what was the key driver, for Affimed to come to TAP and wanted to work with LLS TAP? And also, what was the impact of a TAP agreement?
Yeah, thank you, Chin. I'll do that. And yeah, I remember the very first days of our contacts. Still being a private company at that time, tiny, as I said, maybe 20, 25 people, having this innovative product. Indeed, pretty much at the time also when we had seen that a CD30 targeting agent was very successful. I'm talking here about Adcetris or brentuximab vedotin that was just approved. And everybody saw the very high response rate of this antibody drug conjugate. And the other part of the world was indeed focusing on T cells. So when you as a small company come with an innovative idea, as I explained, going for the activation of innate immunity and trying to help the patient with a very different vision, meaning that if you activate innate immunity, you may also get the benefit of activating adaptive immunity. That's how any immunologic process started. And that was our idea. As you can imagine, it was novel. People were very excited about Adcetris. Finding a partner in the US simply gave us, for the first time, that anybody paid attention to AFM13, because when a patient organization and the society is really supporting such innovative treatment, anybody else will take very much a look at that. So the understanding on your side how important innovation is helped us a lot. As you said, this was almost 10 years ago when we came together, and it became very clear what your vision is, that you were also not just looking for innovative treatments, more effective treatments, but safety was of a particular importance, as we know that most of the lymphoma patients still, including also Adcetris, are only receiving chemotherapeutic agents. You helped us not only to put this on the landmark in the US, it gave us a lot of visibility in the entire medical community, but also industrial community and investment community. So this is how we started our dialogue, and we, through your funds, that indeed represented probably a significant amount of our overall funding. As I said, with 25 people, your expenses are pretty low, yet at that time running maybe 2 clinical programs. That helped us a lot, so we could move forward with AFM13 first in the phase 1 study, but then we were spreading into 2 different phase 2 studies, one in Hodgkin and one in T-cell lymphoma. And on top, we could then start our first combination study. And just let me remind you, AFM13 is active in Hodgkin lymphoma. It can produce single agent responders, both PRs and CRs. And it most recently also showed single agent responses and is very well tolerated. And with, in particular, the combination study that we conducted with pembrolizumab, we were for the first time able to show that a modestly active single agent AFM13 could really push the efficacy of pembrolizumab. This pembrolizumab in itself shows about a 20% CR rate, and we treated 24 patients and ended up with a roughly 45% CR rate. So you can see that you are helping us to set the foundation for many other innate cell engagers today, because we were learning broadly how these drugs work, but also in terms of what you can achieve in combination. So in principle, you helped us to validate AFM13 in order to become a much bigger company what we're today.
Thank you, Adi.
Thank you, June. Can you hear me? Hopefully.
Yes.
So the next question is really directed toward Pavel. Pavel, congratulations again on the activities in late 22 with respect to the partnering deal and the financing. If there are any specific comments that you'd like to make related to your experience, I know that there was some period of time before initiating with TAP formally, and then you worked both with a preclinical focus in addition to the current clinical focus with the TAP team.
So actually, I heard for the first time about the LLS at, I think, BioEurope in Milan, so like, I think back in 2013 or something, when I was by chance sitting against Lou De Gennaro at the dinner. It was a completely chance meeting at the conference, and we started chatting, and he told me about the LLS and the activities of the foundation. And I said, you know, we are still at the preclinical stage with our PIM3 kinase inhibitor, but we may have something for this collaboration, with you in like maybe 3-4 years from now. And this is how we parted. And then, you know, we actually started chatting about, you know, Cell24 with the LLS, but in the meantime, we partnered the drug with Menarini, but we built the connection with the LLS. So when we came up, we're coming up to the clinic with our second program, the CDK19 inhibitor. It caught also the scientific interest of the LLS, because it was also involved in the regulation of RANKS1 pathway, which was of interest to the LLS. And obviously, acute myeloid leukemia as the most serious unmet medical need in all heme malignancies was the top priority for the LLS. So the novelty of the mechanism targeting RNA transcription, forcing cells into differentiation, and also the interesting publications in which we showed how we are able to rescue anemic phenotype with the drug and stimulate erythropoiesis, in addition to killing blastic cells, was also something that drove LLS to kind of be interested in science. So we signed a partnership with the LLS back in 2017, when RIVO120 was still preclinical. When our company had a different name, Cellvita, it was still Cell120 rather than RIVO120. And we've been through, a lot of, some roller coasters together. And like, for example, there was a large pharma company which published a publication suggesting that targeting CDK8 is toxic and people should simply avoid it because it's not possible to drug this protein safely. Since that time, of course, with those, like, I don't know, more than almost 50 patients with the drug, and we haven't seen a single DLT, but we have seen single agent efficacy. So we think that colleagues from this large pharma company were simply wrong. But it was good, to have the authority of the LLS as a kind of independent stakeholder and reviewer kind of in the program. We are saying, okay, investors were asking us, where is this publication from a large pharma company? And we are saying, okay, but we are under a CDA with the LLS. They reviewed all the data and they just paid us another milestone. So what's more important? We also had some, we got a lot of good advice regarding, how to develop the program. So there was a drug that was not so much known at that time called venetoclax. A few years ago, when before it became a kind of household name, before it showed its full potential in the LLS and in AML, and LLS was of course one of the early supporters of venetoclax, not only at the kind of business but also at the scientific level. So they told us, guys, please, this is the big thing that is going to happen, change the AML landscape. So please make sure that you position RIVO-120 correctly in a combination or as a follow-up therapy to venetoclax. So that's the kind of advice also that we've been getting from the LLS. And the third part, I think, connections. RIVO, of course, comes from Poland. We didn't have many US connections. So, creating the first meetings with the potential key opinion leaders, bringing us to the US, investigators was also a very important value added from the LLS that we've received.
Thank you, Pavel, for all that. Back to you, June.
Okay, thank you, Javid. Thank you, Powell. And so thanks for Adi and Powell for sharing their stories of their experience working with LLS-TAP. I'd like to now turn to Marie-Louise. So Ferring became a TAP partner in June 2022, so a little more than like half a year ago. So though it has not been very long, but I want to ask you, what was your experience working with LLS-TAP?
Thank you very much, June. So it's interesting to hear that we are like young in the game, because I feel like we have been collaborating for a long time. But anyway, as I know from my team members, because I wasn't on board, the first meeting with LLS and Ferring was in 2021, I think, at JP Morgan. So at that time, we were like a solid tumor company developing BAX in solid tumors, but we really wanted to go into blood cancers. And like, because of the rationale of the single agent safety and efficacy, and also the CLEVER-1 positivity of the myeloid cancer cell. So for us, it was very helpful to liaise with you, and we got financial support, but very importantly, you helped us to transition from a solid tumor company into like a blood cancer company. So we got a lot of advice on protocol writing, study design, and furthermore, a lot of networking to get into contact with investigators, key opinion leaders, and so on. So approximately 1 year later, last year in June, we started enrolling patients in our phase 1/2 study with BAX in blood cancers. So this is a combination study where we're adding baxmarilimab to azacitidine, a doublet, in patients with refractory AML and MDS, and also frontline MDS. And we have a triplet where we add baxmarilimab to azacitidine and venetoclax in frontline AML that do not tolerate chemotherapy. So we started in the doublet to evaluate the safety. And after the 5 first patients were treated, we saw no DLTs, no safety concerns. So at the dose escalation meeting, we could increase to 3 mg/kg, the next dose level in the doublet, and also open up the triplet. More importantly, but very interesting, I would like to say that 3 out of the 5 patients in the first doublet dosing cohort had objective responses. The first patient, refractory AML, immediate prior treatment azacitidine. After 4 cycles of treatment, complete remission of the blasts in the bone marrow and in the blood, not complete blood peripheral blood recovery, but a CRI. A few weeks later, he also recovered in the blood. So very nice response. Second patient, refractory MDS, after 2 cycles of treatment, partial response. So of course, with the combination treatment, it's always very difficult to know like which of the components of the combination is actually helping the patient. But if you think about it, both patients were refractory to azacitidine. One of them had it immediate prior to the doublet. So I do believe that it's likely that BAX helped to these patients. The third patient is a frontline MDS that had a complete response. So all these patients were enrolled in Finland. But thanks to LLS, we are now ready to open up sites in the US. So I mentioned to LLS, we want to open more sites in the US. Within 2 weeks, we had lots of telephone conferences on our calendars. And within a few weeks, we had all the sites we needed for expansion into the US. So I would like to say that for us, bringing BAX into blood cancers, we could speed up the development immensely by the collaboration with you. And of course, we want the treatments to come out to the patients as quickly as possible. So thank you very much for that.
Thank you, Marie-Louise. Thank you.
Great. And now some questions for Pierre. Pierre, ImCheck is also one of the newer CAP partners, similarly since June of 2022. And when the collaboration started, your lead asset was already in the clinic for solid tumors, is my recollection, and had just entered or was upon entry to the clinic for liquid tumors. So perhaps you can discuss or describe the value of working with LLS, and given that drug discovery and development is expensive and raising capital very important, the impact of LLS participating in the round or helping you and your investor base get comfortable with the liquid tumor space upon closing of the round.
Yeah, thanks. So I think that's exactly right. Initially, our work and center of attention was really solid tumors. We've never abandoned the idea of hematologic malignancies, but as a small company in the process of raising money, and don't forget that raising money, COVID during the COVID time was kind of a challenge, and in the beginning of last year was also a challenge given the market conditions and the interest of the investors or trying to protect their interests. We really felt that it was the right thing to focus on the right thing. Nonetheless, our investors believed, and so did we, that we should not abandon hematologic malignancies. And indeed, while we were in the process of progressing through the Series C, and that we got to know each other, and you got interested in joining, we felt that it would be a great idea to have you as a strategic investor. And we do not only choose our investors because of the money that they bring to us, they need to bring more. So we're not taking opportunistic investors at ImCheck. We're only taking strategics that they are purely financial or present other type of strategies. So we have corporate investors such as Pfizer Venture, but we felt that it would make a lot of sense to bring you on board, help us, and you have, with some of the US network that we're lacking, and I mean investigators, centers, even though it's been a challenge to recruit patients in the US lately with the COVID and the turmoil, but it's getting better. Attract the attention of other investors, not only US but also European ones, after you've raised your hands and wanted to join, but also pharma. I think what I'm expecting from as a benefit is also our ability to be able to advertise the fact that you've chosen ImCheck as one of those 2 to 4 investors that you select on an annual basis, according to Laurie, to convince those pharma that there is something of great value at ImCheck, and then to get in touch with us and collaborate with us, and that is happening. So again, this intent that we had at the beginning to bring you on board for strategic reasons is clearly paying. And I hope that we're going to be able, like Adi and Pavel, who have been now experiencing a long-term relationship with you. Marie-Louise and I certainly want to see that happening for us too. And we certainly count on you. So for us, this is clearly something that if I had to do it again, I would not hesitate, clearly.
Thank you, Pierre, for that. Back to you, June.
Thank you. So Javid, so you went to San Francisco last week for during the JP Morgan Healthcare Week. So besides all the rain and storm that catch the highlight, so what was your highlight from going there besides the weather?
Yeah, thank you for that, June. In spite of 2022, which was a very busy year for CAP and maybe not so good year for biopharma companies generally, but doing well for our partners at least. We had an opportunity to meet with our TAP partners, understand the investor climate, and see what might be similar or different for 2023. So my question to the panelists are, whether you were there or not, how are you finding the current market environment in 2023? Is it better than 22? Where do you go from here? And what are you hearing, either yourself or your team coming out of JP Morgan? So maybe Pierre, you want to start?
Sure. We were not at JP Morgan this year. We have raised close, as a million plus 6 months ago. We have a 4-year runway ahead of us. We talking to investors, US crossovers in particular, almost well, not every day, but regularly. We have a very solid network of pharma. I am an ex-big pharma guy. Our chief business officer is based in Boston, and he has constant connections and dialogue with pharma. So there was really no real use for us to go this year. Heard that the weather was not that attractive anyway. But more seriously, I can tell you 23 cannot be worse than 22 from a fundraising standpoint. And we're glad the year is behind us. I think and I kind of agree with what I'm hearing and from the bankers and the universe of consultants and so on. I don't expect anything to become truly much better before 24 and possibly even the end of 24. And that's not good news, but I think companies need to be very careful with this situation. I am predicting that many of the private biotech and even possibly the public ones will face tough times refinancing. When we look at also the universe of the publicly traded biotech in the US, and I hope that my colleagues over here on the call will be will not face this, but it might be challenging. So all in all, we need to continue to talk to investors on a regular basis, be close to bankers, and convince the world that what we're doing is necessary, and that they trust in our science and results, so that we can survive this this very tough moment. But let's get ready for another, at least in my opinion, 18 months of a complex situation from a financial standpoint.
Great, thanks. Marie-Louise, I know that your team were there. Any specific comments in terms of the key takeaways?
Well, so my team was there, and I agree with Pierre that it couldn't be worse than 2022, but I do have a little bit more optimism than you, Pierre. I do believe and hope that everything will be better by maybe in a half a year, by the end of this year. And I think the numbers of IPOs really are going to increase. So that's what I'm hoping for. That's what was conveyed to me.
Yeah, great. And Pavel, I know you've spent most of your holiday season probably recuperating from your activities of November and December, which were very successful. Any comments you'd like to make?
So this year, only Watnek Vatho, our US-based chief business officer, participated on behalf of RYVO. He had something like 15 banking and investor meetings. And of course, there are some investors interested in RYVO 120, and we are in dialogue with them. We've been doing our IR outreach, but I think that generally there is a little bit skepticism right now towards the biotech sector, and I think that before US funds start massively investing in European companies, especially from maybe less known locations such as Poland, they have to start first investing in domestic companies. So I think that we are well funded. We are focused on execution over the next 2 years. And if the US the situation in the US for US companies improves, then we may consider targeting US investors with some capital fundraising more actively. But obviously, the priority in 2023 is to rather generate more data than raise money in the US. And I think it's a fortunate situation for RYVO. We are relying mostly on our domestic investor base, which has been very loyal, very happy. And since the first listing, they've made like 25 times on their investments, so they still cheer for us.
Great, thank you. And Adi, I know that you were there because Laura and I literally bumped into you at the Pfizer reception and had a chance to chat for a few minutes. So maybe you could provide a unique perspective, both from a fundraising perspective and partnering as well with big pharma or big biotech.
Yeah, I mean, it was very rewarding being back again and meeting those people in person that you haven't seen for a long time. Video is giving you some sort of communication, but the human touching points, it was very important to go through that again, and you take back a lot of good feelings and a lot of good emotions. We had exceptional busy JP Morgan. We had far over 30, almost 40 investor meetings just. So I think that's even not sufficiently summarizing it. A few other analysts we were meeting. So in the 3 days that I had, I hardly left my room, so the weather wasn't bothering me too much. And that was the advantage of the many meetings. Overall, I realized that the sentiment of investors is still to be on the cautious side, making prediction in the future of what it is, but is difficult, okay, for all of us, because I'm always saying if I know what would happen in 3, 6, 9, 12 months, I would buy shares. It's very easy. I would short them. So to make these predictions, what's different? The sentiment is there to invest again. That's very clear. Investors are still very selective. We are realizing it on our end very much. And we have, as I said, presented data at ASH about a combination of AFM13 with allogeneic NK cells. I told you about this very unprecedented response rates. Early, basically, durability data is still early, but just think of these patients. If you wouldn't treat them, they're probably dying in a matter of weeks and months. So these are exceptionally sick patients. All the patients that we treated did not respond to the prior therapy. So that's pretty dramatic. They had 7 prior lines. And we bring this very high response rates. And what's most rewarding for me to see is that the very first patients that we treated, he got on a transplant, and that's now almost 3 years ago, and he's alive. We're taking patients out of hospice. And what we're also learning is that we cannot produce enough of these NK cells in order to fill the demand of treating all the patients that are requesting a treatment. So there is definitely something going on. It looks all very bright for Affimed. Now, where investors stumbling? I said we have used an academic product. So the NK cell is used at site. We have to apply it fresh, and hence you produce small quantities. It's expensive, but you also cannot ship it to other sites. So for us, the transition from this product, which is cord blood derived, over to what I call a product produced at commercial scale. Again, a cord blood derived natural killer cell is a major step. We are very privileged to be able to work with the company that I mentioned before, Artiva, and investors are now sorting out if we not only would get an IND approval, but how fast this new study is to be conducted. We're pretty optimistic. We have received very high-end feedback from FDA and now translating this feedback from the pre-IND meeting into the IND filing, which we're projecting to submit in the first half of this year. And I trust that this can bring back a lot of investors into our stock. As I said, with meeting with like 40 investors, also some banks. On the other side, we have not entertained many interactions with pharma companies on AFM13. It's been a product more on the niche end. In the beginning, a single agent. Now it's particularly with the NK cell combination looks very differentiated, so can address a very large market opportunity. And the pharma companies have shown or are showing now a great interest and are asking us what our partnering opportunities are. We're reasonably well funded, I would say. We have money at least to the beginning of 2025, so we can drive our programs forward in a reasonable manner. That's what we currently focus on, but we are in parallel entertaining these discussions. So it was more than worthwhile to show up and meet at these receptions, making the contacts again and being able to exchange exciting data, also talking to other companies that have made huge progress.
Great, thank you for that. And June, I think we have a few more questions with respect to Europe visa versus the United States.
I think in the interest of time, I'd like to move to the Q&A. So in the panel, and just give a chance to see whether we have a question from the audience. So Blaine has, my colleague Blaine has been monitoring Q&A. So again, so Blaine, so do we have any questions?
Yeah, there's several. Let's see. So one of the questions, I know a few of the panelists have mentioned this briefly, but I think even though each company is based primarily in Europe, I think if you could quickly go around and say what your current U.S. presence is, if you have any operations or personnel located in the U.S., and also any future plans to expand would be helpful.
Okay, I'll start with Pierre.
Okay, great. So yes, we do have a U.S. subsidiary. We do have a presence through our Chief Business Officer based in Boston. And we do recruit patients in U.S. centers, actually in different parts of the country, MD Anderson, Yale, Moffitt, and others. It's been very, we started our U.S. expansion at the time of COVID, so we really had to dial back very quickly. But now that things are getting better, we are going to continue to strengthen our presence down there with probably people and obviously more relationship with investigational centers. So that's the plan, absolutely.
Okay, Marie-Louise?
Yes, so Ferring has an office in Seaport, Boston, and we've had this office for more than 1 year. I'm based in Boston since 10 years, and the head of clinical operations is based in the US as well, and we are hiring primarily in the US. So I would say that 20–25% of the staff right now is US based. We do have fantastic collaborations with MD Anderson, Dr. Naval Daver, and we are expanding our US sites. Next week, we're opening a site in the blood cancer study, and we're opening 2 more sites later on in the US. So definitely, we are enrolling a lot here in our other solid tumor study, and we are building our presence as we speak. It's a priority for us.
Okay, Powell?
So actually, most of our trials and the first trials have always been in the US. So we've had collaboration with several clinical locations in the US, such as MD Anderson, for example, Sarah Cannon, and many others, and both for Cell24 and Rival 120. These are the sites which we are enrolling. Of course, many of our key advisors, such as Dr. Michael Savona, for example, from Vanderbilt, are based in the US. And we are also planning to initiate new IITs in the US around MDS MPN. So that will bring us closer also to some of our, new key opinion leaders. And obviously the phase 2 studies of Rival 120 will be heavily based both in the US and in Europe. We don't have a corporate entity, but as I mentioned, our Chief Business Officer, Vatnak Vatho, is based in the US. So he's our kind of first employee here.
Yes, he's based in Aspen. Yes.
Yes, yes.
Okay. Adi?
Yeah, we brought him to Aspen, by the way. Yes, we're operationally, as I said, we're a US listed company, so we have operations in the US since 2014. Two of our executive members, so 2 out of 6 are US based. Chief Business Officer / Commercial Officer, but also CFO is either New York or Philly based. We have teams of roughly each time 10 people in either New York or Philadelphia, supporting business development, but we have a lot of market analytics, pre-commercial activities ongoing that we, for example, brought in from Insight, and then we have a larger clinical operations team in the US. So everything is important. So being active in the US is important, as we also focus on European sites, dependent on where particular KOLs, let me call it like that, but specific leaders in the space are active. So we're well supported. Our study that I mentioned before, where we combine AFM13 with NK cells, will be predominantly, or at least initially predominantly conducted in the US. Main reason is the NK cell currently is having an ID in the US, but not in Europe. AFM13 could be investigated on both sides. We have high interest at the moment. Still, having said that, we will again work closely with, and I think we're already in contact with LLS on supporting the patient recruitment. And that could become a very exciting study for us. If everything goes well with FDA, it could be a smaller single-arm study that eventually may lead then to a registration in the US first, so that these patients that I described before can have benefit of a novel therapy. And for us, it's not only important to consider the tumor of the patient. For us, the person matters a lot. If you look our vision, we talk about stopping derailing patient lives. We know what cancer means when it's diagnosed. It's not a real disease of a single individual. It's a disease of an entire family. I myself have worked for many years in rare diseases. Again, that's affecting entire families because usually you have no therapy. So that's how we consider cancer as well, that we want to bring back these patients to live a normal life amongst their friends and families. With what we have been seeing so far, we have a great hope, yet we have to now take the next steps, convince FDA to allow us to do this study and support us, and then do the other obvious steps, take the steps of registration. So there's a lot going on in the future in the US, not just on the clinical side, but also on the regulatory side, on the business assessment. So we have a broad set of activities ongoing, and we still see US moving faster than other countries. And that's obviously very important that you can take advantage of this. And we're greatly honored that we from Europe can tap into all these resources, including, I must say, the major funding of our company is coming from US investors.
Yeah, this is great. Thank you. So Blaine, do we have one last question? Because we're close to 1 o'clock already.
Yeah, I think one at a time. I think we'll endeavor to answer any of the questions. We'll ask the panelists and respond to the people that ask the questions.
Okay, great. Okay, so we'll address those questions individually. Thanks again for all the panelists for great introduction and a great discussion. And thank you for taking your time. So we hope you will be very successful in developing your therapies in the future. So now I want to invite Laura to say a few final words. Thank you.
Yes, I want to add my thank you. Also thanking you all in the audience for joining us today. And I hope you've enjoyed this discussion as much as I have. Again, we're very actively investing. We're always looking for innovation. We hope that we've inspired some of you to get in touch with us and make us aware of what we're maybe missing. So we are really just an email away. And please also consider supporting LLS through a donation, not just this work, but the many, many activities LLS is supporting for patients, for their families, as Adi pointed out, many, many activities that really can only continue if you help us. And last but not least, we really hope to see you back for future events, and we wish you a wonderful and productive rest of the day. Thank you.
Thank you very much.
Thank you.
Thank you.